everyone, and welcome to my talk on studying Glycan 3D structures with PyMC and ArviZ. My name is Agustina and I work at the San Luis Applied Mathematics Institute in San Luis, Argentina. I am a PhD candidate in biology. I am near the completion of my PhD, which I have done with a full scholarship from the National Scientific and Technological Research Council, which is the government agency for scientific promotion here in Argentina. I have participated in the Gold Summer of Code program, both with PyMC and ArviZ in the past. And I would like to encourage grad students or even undergrad students to participate in this program. It is a great opportunity for learning and self-improvement overall. And recently, I have been incorporated to the core contributors team at ArviZ. So this is the outline of my talk. Probably the most interesting point for the audience of this talk is the Bayesian model for exploring Glycan 3D structures. But as this model incorporates a lot of domain knowledge about Glycan structures, I must introduce a few other concepts first. So I will start with Glycans, then I will talk about torsion angles and how they relate to Glycan 3D structure. I will dedicate a few minutes to circular variables, as torsion angles are. Then I will move on to energy and the Boltzmann distribution. And finally, I will combine all of these concepts together in the Bayesian model for exploring Glycan 3D structures. So, what are glycans? Glycans are one of the groups of molecules that constitute living beings. The other three groups are proteins, lipids, nucleic acids. Glycans play numerous roles in cell structure, signaling, disease, infection, and many others. They consist of single units linked together, as you can see here. All of these monomeric units are connected. When present in foods, glycans are commonly known as carbohydrates. So they are present in the foods that we all love to eat. Some other examples of glycans are the structural components of vegetal cells. And most recently, glycans have been described in the spike of the SARS-CoV-2 virus. Here we can see a full atom representation of the spike in the SARS-CoV-2 virus. And on the right, the blue parts represent the glycan portions of the spike. So we can see how big and important they are. Now I'm going to tell you about torsion angles. For that, let's take a look at this glycan molecule. It is a small one, as it only has 2 subunits. And in this representation, every atom in the molecule is shown. But for a moment, let's focus only on these 4 atoms that I have highlighted here in orange. Notice that these 4 atoms are on the linkage between the 2 subunits. The reason for only accounting for these 4 atoms is because 4 atoms is the minimum number that we need to define a torsion angle. So torsion angles are angles between 2 planes. And as you can see here, these 3 atoms are defined in plane beta. And at the same time, these 3 atoms are defining plane alpha. And the angle between them is their torsion angle. In this animation, you can see that as the torsion angle changes, the three-dimensional disposition of these atoms will change, as well as the three-dimensional disposition of all the atoms connected to them. In that sense, a glycan's three-dimensional structure can be uniquely determined by the values of the torsion angles in between their units, and that will be really important for this talk. As you may have noticed, torsion angles are circular variables. It means that they can be represented as angles over the interval minus pi, pi. And their density F will take the same value at both endpoints. Circular variables have an inherent complexity to them. When working with them, we must account for the curved nature of the sample space. For example, if we compute the sample average of two circular data points, for example, minus pi and pi, represented here on this circular plot, we can see that the vector average, that maybe is the most intuitive thing to do, is completely outside of the sample. The correct way to do it is computing the circular average. Increasing support for circular variables was one of the objectives for my last Google Summer of Code project with ArviZ. And in that project, I modified ArviZ plots like the distribution plot or the trace plot to adapt to a circular setting. Also some functionalities like the ArviZ summary. You can find these new features on ArviZ's latest release. Now is a good time to introduce the von Mises distribution. You can think of the von Mises distribution as a normal distribution wrapped around a circle. It has two parameters, mean and concentration. And when the concentration approaches zero, we get a uniform distribution. And this may be more apparent on the linear representation on the left. As the kappa parameter increases, the density will concentrate around the mean. We use the von Mises distribution as a prior distribution over the torsion angles on a glycan molecule. Actually, we use a multidimensional von Mises distribution as a prior with as many dimensions as torsion angles are in the glycan molecule. The prior parameters mu and kappa are set according to prior knowledge about glycan structure. Now, a few words on energy. Energy is defined as a body's ability to perform work due to its position relative to other objects, electric charge, and other factors. For explaining why we care about energy, let's recall those four atoms that defined the torsion angle earlier. What you need to know is that different torsion angles will result in different glycan structures. And as the torsion angles vary, the energy of those structures will change too. In a given molecule, there are torsion angle combinations that will cause atom superpositions or clashes. Those particular structures will result in very high energy. The Boltzmann distribution describes the probability of a system being in a particular state as a function of its energy and temperature. So this distribution relates the energy and temperature of a system through the Boltzmann constant. According to this, at a fairly low temperature, those high energy states I mentioned earlier will have low probability. And only as the temperature increases, high energy states will become more likely. Okay, so In this slide, I will try to summarize the key concepts I have talked about. These are the ones you need to understand the model. So far, we have learned that glycans are formed by connections between single units, that their connections define torsion angles, and as they are circular variables, they must be modeled correctly. We know that torsion angles define the three-dimensional structure of a glycan, and that certain torsion angle combinations will result in high-energy glycan structures. And according to Boltzmann's distribution, those high-energy structures have low prior probability. Okay, so now we can move on to the Bayesian model for exploring glycan 3D structures. I am actually going to show you two different models. The first one is very simple, and in the second one, things will get a bit more complicated as we add experimental and theoretical data. But for now, let's focus on this diagram of model 1. We have the prior probability on the torsion angles of a given glycan molecule. Then we compute the energy and the potential. This Bayesian model is part Bayesian model, part simulation. Because as I tried to represent here on the right, there is a simulation of a glycan molecule, and these glycan molecules' torsion angles are set to the values that were drawn from the prior. The energy of the resulting structure is computed, and then the potential. PyMC3's potential allows us to assign higher probability to low energy structures. So in this model, the only restriction the sampler finds is that of the energy of the proposed structures. We tested this model on a glycan molecule of known structure. In this way, we can validate our model. This glycan has 3 subunits and 4 torsion angles. We can see here the trace plot for a run of model 1, and we have the posterior density of the 4 torsion angles in this molecule, and also an estimate of its energy that derives from the potential. We solved this model with PyMC3's sequential Monte Carlo. If you are interested in sequential Monte Carlo, you can watch the PyMCAL talk, Sequential Monte Carlo, Introduction and Diagnostics by Pedro Ramirez and Osvaldo Martin. On this trace plot, we can already see where most of the density for these torsion angles is located, but this representation here is a Ramachandran map and is the standard representation for torsion angles when it comes to biomolecules. Here we have 4 torsion angles separated in 2 pairs, and these areas represent the most visited states by the glycan molecule. They are colored according to their energy relative to the energy minimum. As I mentioned earlier, the structure of this glycan is known, so we have experimental data on the torsion angles to validate this run. As we can see for the first pair, the energy minima and the experimental data do not match as they do in the second pair. This may be due to the fact that in our simulation, we only accounted for the glycan molecule, while in the experiments, the glycan molecule was bound to a protein. And finally, in this animation, we have posterior samples represented as glycan structures. it is important to notice that all the structures are in agreement with Boltzmann's distribution, in the sense that most structures we get do not have atom superpositions, even though probability of such structures is very low, but non-zero. So for model 2, we're going to add data. If the structure of this glycan molecule is known, that means that there are experimental data available. And this experimental data does not consist of direct measures of torsion angles, but indirect observables related to them. So we can incorporate this experimental data in our model alongside with a software that provides theoretical counterparts for these observable quantities. That software is called GSoC and was developed in a research group. This is model 2, and part of the model is the same as model 1, except that for every proposed glycan structure, we will compute the theoretical observables and perform a linear regression. That means that the x variable in this linear regression is not fixed. It is changing in every iteration of our sampling algorithm. For the likelihood, we use t-student distribution because things can go wrong during the theoretical data computation. So we need our model to be a little bit more robust so that these anomalous theoretical values do not complicate sampling. And we have our prior probabilities on the rest of the parameters. Model 2 showed similar results as model 1, but sampling is a bit easier for the sequential Monte Carlo due to the inclusion of the likelihood function. And again, let's use Ramachandran maps and animations to explore the results of model 2. The results are very similar and lead us to the conclusion that even if the linear regression model does not imply better results, it helps with sampling. And this is valuable because as the posterior distribution will become more and more complicated in a larger glycan molecule, this can really make the difference. So this is the end of my talk, and please contact me if you have questions or comments about this talk. The work I presented here today was done in collaboration with Osvaldo Martin, who is a core contributor at PyMC and ArviZ. Here you have our email addresses and Twitter handles. Don't hesitate to interact with us and contact us. We would like to know your impressions on this work, and thanks for watching. Bye.